Uh, we will do an experiment to examine starch or potato particles under the microscope. This experiment, there is independent variable, dependent variable, control variable. Independent variables include concentration of solution, which we put the potato particles in it. Okay? Dependent variables, which mean or like the changes occur in mass of the potato before and after the experiment. Understand? Control variables, which we must keep it constant during performing the experiment, like volume solution of solution, temperature, time, surface area of the potato. All these control variables should be constant. Okay?
Okay.
Culturing microorganisms in the lab use agar jelly, which contain nutrients. This is agar jelly, which is a nutritive medium. We put the microorganism on it to grow and multiply. The bacteria will form colonies on the agar used in curating fluids to add the bacteria to the agar jelly. In a school lab, the microorganisms are kept under 25 degrees centigrade to prevent the growth of any harmful bacteria. This is condition we use to grow up the microorganisms. We use suitable temperature and nutritive medium. Okay?
Okay. Okay.
Investigating the effect of antibiotics and bacterial growth. We put the antibiotics on the colonies of the bacteria we cultivate in the agar jelly. Place paper like paper discs that have been soaked with different antibiotics on agar plate that has bacteria on it. The antibiotics should diffuse on the agar. The most effective antibiotics at killing the bacteria will have the largest inhibition zone. You understand what I mean by largest inhibition zone? Inhibit it, the most effective antibiotic will inhibit the growth of the bacteria. You understand? I will draw for you what we can. This is the test and the growing bacteria on it, like that. You see my drawing? Yusuf.
Yeah.
You see my drawing?
Yeah, I could see it.
Okay. We put here different antibiotics. Like here, 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 here. These are different types of antibiotics. Antibiotics which have more effect upon the bacteria will make largest inhibition zone, which means the zone that bacteria cannot grow like this. Okay? like here. This is a smaller. This is a smaller one. And if you're the lab doctor, you will write your report to the doctor or
Okay.
physician. You will agree مثلا A, antibiotic A, antibiotic B, antibiotic C, antibiotic D, antibiotic S, antibiotic R. You will write the most effective antibiotic is the antibiotic A, which has the
Mhm.
largest inhibition zone upon the bacterial layer. Understand, money?
Okay, just a question. Is it fine if I quickly go to the bathroom? I'm really sorry. I
Okay,
forgot to go before. Just really
okay,
quickly, I'm sorry.
okay.
آي. أنا آسف جدا. أنا آسف جدا. أنا 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 بس أسألك حاجة بس.
اتفضل.
يعني الانهبيشن zone، يعني is like the place it can attack the bacteria, or the the amount of bacteria it leaves behind.
Inhibition zone means that the zone where the antibiotic work on the bacteria, which we grow it on the disk. So the antibiotic can kill the bacteria, so inhibit its growth.
So when
Understand?
you put the antibiotic onto the bacteria, it
Yes.
will kill as much as it can, and then it will stop, right?
Okay. Good.
Okay.
Yeah.
Thank you.
Okay.
Thank
In each experiment, we should use control. Control, which mean we use something cannot change the parameters or the real parameters of the experiment. Understand?
you. Hmm.
The
I
control
understand.
which we use in each experiment, if this is a substance or a chemical, we use it, which cannot change the standard parameter of the experiment. In this experiment, we can use sterile water. Sterile water will not affect the bacterial growth. So we
Hmm.
consider it as a control. You can make a matching between the normal growth rate and the effect of different antibiotics we add to the dish.
Hmm.
Understand?
Understand.
Almost the cultivation of the bacteria was an incubator for 48 hours at 25 degrees centigrade to leave them to grow enough, then adding the antibiotics.
Hmm.
Any question?
Hmm.
You have any question?
No question. Thank you.
Now we will discuss different types of cells. Before discussing different types of cells, we will discuss the types of cells in general. Types of
Hmm.
cells in general, there are prokaryotic cells, and eukaryotic cells.
Hmm.
The suffix pro means no nucleus in
Okay.
this cell. Eukaryotic cell means there is a nucleus, or cells which have nucleus. Good. Let us draw these cells.
Hmm. Hmm.
We
قولها
will,
تاني.
we will draw this cell. You see the board.
Yeah, I could see it.
We will first we will draw the prokaryote.
Hmm.
We say pro means there is no nucleus. Okay,
Hmm.
this is a cell membrane, a cell envelope, no nucleus. genetic material which was the DNA. It was rounded, small rounded, and attached to the cell membrane. And there is some organelles like ribosomes
Mhm.
and some source of energy as mitochondria. No much organelle. This is the cell membrane.
البروكريا البروكرياتيك ملهاش cell wall صح؟
No cell wall. There
مهم.
is some prokaryotes have cell envelope, not called the wall. We will discuss this later.
مهم.
Okay. Cell membrane.
Okay.
This is the DNA. This is the ribosome. This is the mitochondria.
مهم.
Okay.
Okay.
Why should this prokaryotic type of cells have ribosome? To be able to synthesize the protein.
مهم.
Okay.
Okay.
Why it may contain mitochondria? To obtain their energy. Used for chemical
مهم.
reaction or metabolic reaction inside the cell. Good. And why DNA? DNA, this is the genetic
مهم.
material. You can replicate.
مهم.
It is the genetic material. Okay. This is
Okay.
the prokaryotes. Some prokaryotes have an envelope or chitinous coat. Chitinous coat. For
اسمها
protein.
ايه؟
Chitinous.
تايتينيوس.
Chitin or
ده
chitinous.
ايه؟ ده ايه؟
Hmm?
مش فاهم
So,
اللي انت قلته عشان.
some prokaryotes have chitinous or chitin, a coat of chitin type of protein.
مهم.
This
يعني
is
it's protein, right?
yeah,
That's what you said? Okay.
okay, chitin or chitin. This coat
مهم.
for protection from the surrounding changes of the environment as temperature, chemicals, pH value, acidity or alkalinity changes in the surrounding meat. This type of prokaryote will build this chitinous coat to protect themselves. Okay?
مهم. Okay.
Most prokaryotes example is the bacteria. All bacteria are prokaryotes.
مهم.
Okay? This is the prokaryote.
Okay.
Prokaryote and eukaryotic cells. Eukaryotic cells have membrane-bound organelles as, for example, plant cells, animal cells, and fungal cells. Prokaryotic cells don't have contain a nucleus, for example, bacteria cells. Okay?
مهم. Okay.
We will discuss the difference between eukaryotic plant and animal cells.
مهم.
plant, eukaryotic, like plant, cell, animal cell.
بس سؤال، بس اليوكاريوتيك اللي هو اللي عنده السيل وول صح؟
No, have a nucleus.
اه يعني
Eukaryotic
اسمها إيه التاني
have a real nucleus.
مهم.
البروكاريوتس
مثال
don't have a nucleus.
البروكاريوتيك does not have nucleus but اليوكاريوتيك has nucleus.
Has a nucleus.
Okay. And the prokaryotic is usually bacteria and other cells, right?
Mostly bacteria. Mostly.
Mm.
Almost
Most, and
bacteria.
then eukaryotic is all the living cells like animal cells and plant
Yes,
cells.
it is more developed cells, type of cells.
Mhm, I get it now.
Pro mean primitive. You understand what the meaning of primitive?
Mhm,
Primitive type of cell. Primitive. Primitive.
mhm.
Primitive type of cell. Okay?
Okay.
Roll types. Eukaryotic cells, we say like plant cell, animal cell, algae, and fungus.
Mhm,
Algae, and fungus. We will discuss the difference between plant and animal cell. Okay? Plant cell almost has definite shape. Almost have definite shape. Animal cell
mhm.
have a regular shape, round.
Okay.
Okay? This definite shape because the presence of cell wall. Presence of cell wall.
Mhm.
Okay?
Okay.
Underneath there is the cell membrane.
Mhm,
This is the cell membrane.
mhm.
There is no cell wall in the animal cell. Just cell membrane.
Mhm.
Okay?
Okay.
Eukaryotic characterized by presence of membrane-bound organisms.
Mhm,
Membrane-bound organisms.
mhm.
Like we already have here nucleus, and here we have a nucleus.
Mhm.
What is the membrane-bound organism? Like endoplasmic reticulum,
Mm?
endoplasmic reticulum. This is a transport system inside the cell. Endoplasmic reticulum. Right here. Plant cell have a specialized structure for performing photosynthesis. You understand what I mean by photosynthesis?
Yeah, it's how plants make energy.
No, help the plant to make their food.
Oh, to their food, yeah.
Yes.
It's,
Help the plant cell to form
it's
or build up or synthesize their own food.
Yes.
Yes. This is the chloroplast. This is it's called chloroplast. Not found in the animals. This is the chloroplast. Animal cell don't have chloroplast. Okay.
Okay.
Plant cell have a large vacuole.
Mm-hmm.
In animal cell, the vacuoles are small and multiple.
Mm-hmm.
While in plant cell, the vacuole is large, one or two, but they have a large size. Okay. Other
Okay.
organisms like mitochondria found in both
Mm-hmm.
mitochondria. This is for energy production. They
Mm-hmm.
consider the lung of the cell
Mm-hmm.
for respiration. Using the respiration through mitochondria to produce energy. Okay.
Okay.
We go back to our paper. You can read this comparison.
Mm-hmm.
Animal cell have a nucleus.
Animals, animal cell and plant cell have a nucleus, and they both have cytoplasm, and only the plant cell has chloroplast, and then they also both have cell membrane, and only the plant cell has permanent vacuole, and then both have a mitochondria and the ribosomes, but the plant cell only has the cell wall.
Good.
Yeah.
Bacterial cell, which is we you know that bacterial cell is a what type of cell?
It is prokaryotic.
Yes, good. Prokaryote. Bacterial cell don't have true nucleus. They just have single strand of DNA. No, this this
Mm-hmm.
is the DNA. Okay, that flow
Okay.
in the cytoplasm. They contain plasmid. Plasmid. This is the small circular DNA found in the cytoplasm of bacteria. This is
Mm-hmm.
meaning of plasmid. This is the plasmid. Prokaryotic cell produced by binary fission. This is the type of reproduction for prokaryotes. This is the
Okay.
this is the process of binary fission. This is the mother cell. Do the division become two daughters. So
Mm-hmm.
two daughters. This type of division or reproduction called binary division. The cell is
Mm-hmm.
split in two, and the mother disappears. You understand?
Understand.
The mother, after
Yeah.
division, disappears. Its body will split into two daughters.
Mm-hmm.
Good.
Good. Good.
we already know that the cell is the unit structure and function of all living organisms. Structure mean building the body. Function mean do all vital activities of the living organism. Understand what the difference between structure and function? Structure mean building the body, function doing the vital activities. Okay? Each cell has its own characteristic shape and the function according characteristic shape according to its function. Each cell has its own shape to perform its function. Good. Let us discuss how this you understand what I mean by differentiation? This word? Understand what I mean?
No, can you explain?
Okay. When you are in your mother uterus, first days for you in your mother uterus, what did you wear? What is your structure this time?
What is what?
When you are in your first days in your mother uterus, what is your structure?
One cell, right?
One cell, which is called zygote. Good. Okay. This zygote will divide. With this zygote will divide, become some or mass of cells. Good. By time, this mass of cells will differentiate. This word, differentiate into some cells become nervous system, some cells become muscles, some cells become bones, some cells become your eyes, some cells become your hair, some cells become differentiate into the digestive system, circulatory system, heart and vessels, some cells became lungs. Okay, this what meant by differentiation.